In this video, I'm going to talk about DNA sequencing, and specifically I'm going to focus on whole genome sequencing. Whole genome sequencing comes in two flavors. First, there's de novo. This is if you're sequencing a genome for the first time. So here, de novo meaning from new, you want to sequence all of the genomic content in your organism. There's also whole genome resequencing. Resequencing is when there's already a genome for that organism, but you think that there are small variants in your population, so you want to resequence the entire genome and compare it to the reference genome in order to see those small variants. Sometimes people perform de novo sequencing, even if there is already a reference sequence, if they're expecting big changes in their population. Like for example, in bacteria can undergo horizontal gene transfer, that is basically pick up new genes, new sections of genomes. So if you were interested in those big, large-scale genome changes that might happen in your organism, even if there already is a reference genome, you might still do de novo sequencing just to make sure that you're capturing everything. So let's talk about bacteria for a second, and let's imagine that in my perfect genomics world, I want to sequence the genome of this bacterial cell. So what I could do in my perfect world is just zoom into this cell. Say, oh, there's your big circular bacterial chromosome bacterial cell, and I want to sequence it. I want to read all the A's and G's and T's and C's, and so maybe I pick somewhere and I start going along and reading that sequence. And I could just do this linearly for the whole, if this is E. coli, 4 million some base pairs in order to see the entire genomic sequence of that one cell. But as we're going to learn, that's not actually possible. We can't just go look at a single genome in a single cell and read all the way along its entire chromosome all at once. There are a couple of problems limiting our ability to do that right now. We can hope that technology overcomes them in the future. One problem is signal. That is, we just can't see a single base of DNA. I can't look inside that cell and read the genome. You can think of this as the same way that we can't see a single light bulb from outer space, but we can amplify the signal. We can have positions on Earth where there are thousands and thousands of light bulbs, and then we can see them from Earth. We can amplify the signal. We can do the same thing with DNA too. We can't see with our eyes or with our sequencing machine single molecules, single base pairs of DNA, but we can employ techniques in order to amplify the signal. A popular one is based on polymerase chain reaction, or PCR, and the idea here is that you can exponentially amplify DNA. So if you started out with a single molecule of DNA, you can amplify it one time to get 2. So that's one cycle, gives you 2 times the copies. You can do another cycle of amplification for a total of 2 cycles of amplification to get 4 molecules of DNA, and so on and so on. And you do this over and over again until you have amplified your signal so that now we can see it with our sequencing machine. But one problem here is that amplification can introduce a number of problems itself. For example, you can have bias. That is, along a genome, along a section of DNA, certain regions might be more amplified than other regions. We call this bias. And one way to counter bias is to make sure that you don't over-amplify. During a lot of what we call library preparation, that is getting your DNA ready to be sequenced, we undergo things like cycle checks to make sure that we have amplified our sequence just enough so that we can see the signal, but not too much. Because you can imagine, the more and more you amplify here, you're just going to increase this bias. This one's going to stay low, and this one's going to get really, really high because this is operating on an exponential scale. Another problem with amplification, and this is true anytime you're copying DNA, is that you can introduce errors. This is sequence errors. For example, here, if I was amplifying this DNA sequence, ATGC, The enzyme that I'm using, this arrow represents the function of an enzyme, a polymerase. Sometimes they just make mistakes, and here it might accidentally copy that G to a C. And now right here, we aren't going to know which was the correct sequence. The sequencing machine itself, the sequencing process, can also make mistakes. So again, for example, you can have this real template that's A, T, G, and C, but this part, this real template, is invisible to us. We as users do not know this. This is the bit that's hidden inside the cell that we can't see with our eyes. We have to do this amplification and this sequencing process in order to get reads. This is what we do see. We see reads. So one thing we can do, besides using a high fidelity polymerase and high fidelity enzymes to make these, to get that error rate as low as possible, is we read the same piece of DNA sequence over and over and over again to achieve a consensus. That means that even if we did see one base pair here that looks different, we can say, overall, these were mostly G's at this position, so I'm going to assume that C was a mistake, and my consensus sequence for this region is G. Another limitation for DNA sequencing is that we can't sequence very long segments of DNA. And by very long, I mean the millions of base pairs that would make up, for example, an E. coli chromosome. We're still not very great at that. We can only sequence in short fragments. So again, going back to this idea of library preparation, preparing your DNA to be read, it has to happen in a couple of steps. So for example, if you start here with you can consider this to be an entire chromosome of information, there's going to be some sort of fragmentation step where you break it into chunks that we would actually be able to read with the DNA sequencer. But the DNA sequencer needs a couple of other things too. Right now, the ends of these pieces of DNA are all unique, right? These are unique positions along the genome. And in order for the sequencing to work, we often have to add adapters of some sort. These are going to assist us in that amplification process as well. So in this example here, I am presenting this based on one very popular mechanism of DNA sequencing. There are other ways to do this too. But by one very popular method is we use these sequencing adapters. So we have a forward adapter and a reverse adapter, and we put these on every fragment of DNA, and that allows us to amplify it, and it also allows the sequence machine to recognize this as a place where it should start reading the sequence. And again, those adapters and sequences that you add onto the ends of the fragments not only allow the sequencer to read it, but they allow us to do that amplification step. When it actually comes time for the DNA sequence machine to read the template being what I've shown here in blue, and the reads, that is the output of the sequencer machine here being showed in green, there are a few ways to do it. You can do single end sequencing, which is just that you sequence from one side only, and that you would end up with reads that look like this. You can also do paired end sequencing, which basically means that for each region of the genome, you're going to get two sequences, one from the forward and one from the reverse. Obviously, this is going to be a little bit more work. You're going to be having to do a little bit more sequencing to get coverage of the same area, but I'll show you in a second why this might be a good idea in some circumstances. Sequencing an entire genome in fragments makes it hard to assemble that sequence back to the full length. This is often compared to the problem of a puzzle. There's a complete puzzle worth of information, but this is what you get back from the sequencer. It looks like a big jumble of puzzle pieces. It's your job as a genomic scientist to assemble those reads to recreate the full genome sequence. I'll try to diagram that simply here. So here I'm showing you all of the reads. These are short little sections of DNA sequence that you got back from your DNA sequencer, but it's your task to recreate this entire contiguous linear chromosome. The idea here is that you find a region of overlap between the reads to assemble them into contigs. Contigs, or areas of contiguous sequence, are regions where you have found overlap. You may be able to further consolidate those contigs into scaffolds. These are regions that are maybe made up of multiple contigs, but you're still not to the level of the entire chromosome, which you can get just through more computation and finally finding all of the regions of overlap so that you get that full contiguous sequence. Depending on what genome you're using, you might find that if you just don't have enough, sorry, I did not mean to move that. If you just don't have enough reads, you didn't get enough sequencing for your organism, you might find genomes that stop at the contig or scaffold level of assembly, and we're just working on it. When we're using the genome browser, we're going to see full chromosomes worth of information for humans and zebrafish and fruit flies. This is because lots and lots of people have been working on this for a long time. But don't be surprised if you're working on a less studied organism that your genome only has the assembly at the level of scaffolds or even just contigs. This assembly step is where sometimes it's a good idea to have paired end reads versus single end reads. Remember I told you in the human genome, there are lots and lots of repetitive sequences. This is true in other genomes as well. So imagine you have this single end read here, you only read forward in one direction, and you're trying to align it to what in reality looks like this. There's a repetitive sequence, a little gap, and then there's a unique sequence over here. This single paired end read, which I've drawn as a red block, could either be aligned or mapped to this region, or this region, or this region. You just don't know, because it's only a single end read, that's the only thing you have. It matches each of those positions equally well. If you had a paired end read, you read from the other direction as well, you can match these single end and paired end reads together, and now you can see that that sequence with the red block and the yellow block actually maps right here. So this is helping you make some of these assemblies and mapping when you have lots of repetitive sequences. So what does it take to have a good DNA sequence, a good genome sequence? Well, for one, longer reads are better than shorter reads. Longer here, I'm going to say, is maybe thousands of base pairs at this point, whereas shorter reads might be 50 or a couple hundreds of base pairs. This is still a very popular way to do sequencing, even though on the last couple slides that I showed you, it could be really hard when it comes to genome assembly. Also, it's really important for this sequencing method to be accurate. What I'll call accurate is about a 0.1% error rate. This means that you're still going to be making a lot of mistakes when you think about the billions of base pairs that make up a genome. But so far, as of 2020, we're still seeing error rates about this much. There are other methods that are less accurate but still popular for some reasons we're going to explore in class. And by here, inaccurate, I'm going to say greater than about a 1% error rate. Another consideration is throughput. And for a lot of our purposes, for getting entire genome sequences, high throughput is better than low throughput. And what I mean here with high throughput is that you can sequence many DNA molecules at the same time. That is, I can chunk up the genome, and on a single instrument, that is a single sequencing machine, I can be sequencing different regions of the genome at the same time. Whereas some low throughput methods, which we still use today for important reasons, but it would be really difficult to sequence an entire genome this way, is you really can only look at one DNA molecule at a time. So you might be thinking to yourself, wow, when I sequence a genome of interest, I just have to find the one method that has really long reads, that has a really low error rate, and that's high throughput. Easy peasy. But what you're going to see is there actually isn't one great method that is good at all of those things right now. So basically there are trade-offs. You can have long reads and high error rates, or shorter reads and low error rates, and there are also differences in cost. So what we're going to do in class is we're going to compare some popular sequencing methodologies and platforms, and we're going to compare the read length, error rate, throughput, and cost to see why some people might use one tool over another.